Now that we've completed our discussions of glycolysis, gluconeogenesis, as well as glycogen metabolism, we're ready to move into our next chapter discussing the citric acid cycle. With glycolysis, we've looked at how we can begin to oxidize a glucose molecule, converting the 1,6-carbon glucose molecule into 2 molecules of pyruvate. This is an energetically favorable set of reactions that represents a series of catabolic reactions. So we do produce a small amount of net ATP as well as some electrons in the form of NADH. What we want to do now is move on into the citric acid cycle where we can begin to fully oxidize that glucose molecule, extracting as much energy as we can from the glucose. We're now ready to move on into what we would consider cellular respiration. The next few parts that we're going to discuss are considered cellular respiration because these next few steps are linked directly to the consumption of oxygen and the production of carbon dioxide. What we're going to do is we're going to see how we can follow glycolysis that we've already discussed, as well as break down fatty acids and break down amino acids, producing acetyl-CoA. That acetyl-CoA can then enter into the citric acid cycle. With the entrance into the citric acid cycle, we're able to completely oxidize the carbons, releasing as many electrons as possible, so that eventually we'll be able to discuss the electron transport chain, where we can take all of these reduced electron carriers and use those to produce energy molecules. Before we move into the specific steps that make up the citric acid cycle, let's look a little bit at the structure of the mitochondria and the compartmentalization of reactions. The steps that we've discussed so far, such as glycolysis, are all going to occur in the cytoplasm of the cell. Now that we move to the citric acid cycle, we need to look at the mitochondria. As you will recall, the mitochondria has an outer membrane, shown here in brown, and then directly inside of that outer membrane, you have a small amount of space, and then you have another very convoluted membrane called the inner membrane. The area within the inner membrane is considered the mitochondrial matrix. The citric acid cycle, with the exception of one step, is going to occur within the area called the matrix inside of this inner membrane. And I know we're not quite there yet, but eventually we're going to see how all of our reduced electron carriers can move through the electron transport chain. And so the actual enzymes that make up the electron transport chain are going to be inside transmembrane proteins within this inner mitochondrial membrane. And we're going to use the inner membrane as a selective barrier so we can build up concentration gradients. So what we're kind of going into now is looking at what's going on inside of the mitochondria. We've got our pyruvate that was the end result of glycolysis occurring out in the cytoplasm. That pyruvate is going to be pulled across the membranes of the mitochondria into this mitochondrial matrix so that we can see the reactions occur. One of the most important steps we need to do to get ready for the citric acid cycle is convert our pyruvate molecules we've produced into acetyl-CoA. Overall, this is going to be an oxidative decarboxylation reaction in which we're going to take pyruvate, a 3-carbon molecule, and we're going to first remove a carbon and then oxidize so that we can produce our acetyl-CoA. With this particular reaction, the enzyme responsible is collectively called pyruvate dehydrogenase. Pyruvate dehydrogenase is actually a complex with multiple parts. With pyruvate dehydrogenase, we're going to see 3 enzymes working together. And so shorthand, we see E1, E2, and E3. That's our 3 enzymes working together. And then we also have several cofactors, some co-substrates. We have a prosthetic group, a lot of things going on. So we're going to look at what the CoA specifically can do, as well as this thiamine pyrophosphate, the TPP, which is actually vitamin B1. The NAD to NADH, we already know that's going to be the little co-substrate that we can reduce. So the NAD is going to carry the electrons that we pull from pyruvate as we're oxidizing the pyruvate molecule. The FAD and the lipoate and the TPP are all going to be part of the enzyme complex that allows us to channel some electrons through there. So before we look at specifically how this pyruvate dehydrogenase complex works, let's look at some of these coenzymes and cofactors. The first one we want to look at is coenzyme A. Coenzyme A is specifically designed to accept and carry acetyl groups. So very often you'll see any 2-carbon group, if it needs to be transferred from one molecule to another, you'll see this coenzyme A. Coenzyme A has a site here shown in pink. It's a very reactive thiol group. It allows us to produce high energy thioesters. So what we'll do is we'll take this acetyl group that we pull from pyruvate, we'll attach it to the end, and then just shorthand all of this. All of this we'll call CoA, and you'll see this high energy thioester bond forming in between. Another important part of the pyruvate dehydrogenase complex is a prosthetic group that we see here called the lipoyl lysine. It's the lipoyl lysine is this lipoic acid that is attached permanently to a lysine residue in the second enzyme of the pyruvate dehydrogenase complex. The lipoyl lysine allows us to have a site for oxidation and reduction. So you'll see this is the oxidized form with your 2 sulfurs bound, and then you'll see this is the reduced form in which you have 2 thiol groups because they're holding excess electrons. We'll see at one point during this enzyme process that the acetyl group that was originally part of pyruvate is going to be bound here, and then we'll see this other area here is holding an extra electron. So this is going to be part of the middle. This is part of your E2 reaction. With the pyruvate dehydrogenase complex, it's very large. We have the 3 enzymes shown here in blue. So enzyme 1 is pyruvate dehydrogenase. This is going to be the area where our thiamine pyrophosphate, TPP, is bound. From the name, you can see pyruvate is going to be the substrate. It's a dehydrogenase, so this is where we're going to pull some electrons from pyruvate. E2 is dihydrolipoyl transacetylase. This is going to be the portion of the enzyme that has the lipoyl lysine residue, and it's going to be transporting the acetyl group from the original pyruvate so that we can release as acetyl-CoA. And then enzyme 3, dihydrolipoyl dehydrogenase, you can see this is another area where we're moving electrons. Enzyme 3 is responsible for getting all of the original parts back in the correct oxidation state so that the reactions can continue. By linking all of these enzymes together in a complex, we allow ourselves to continually see this reaction occur because we can channel one substrate from one catalytic site to another, eliminate side reactions, and highly regulate everything that's occurring. So now let's look at this total set of reactions. I'm going to flip to the picture, but here I've written in the notes for you so you can keep up with what's going on. Okay. So shown here, this yellow area, that represents your enzyme 1. Green area represents enzyme 2. And then pink area is supposed to represent enzyme 3. So let's start here with enzyme 1, pyruvate dehydrogenase. In enzyme 1, we're going to see a decarboxylation of pyruvate. So here's our pyruvate comes in. We decarboxylate, releasing our CO2. So this is our first fully oxidized carbon that we're releasing from our original glucose molecule. The TPP is part of your enzyme 1. TPP serves as an acceptor of your acetyl group that came from pyruvate here. So TPP kind of acts like a swinging arm. It can then bring your acetyl group directly to interact with your second enzyme. Now this acetyl group is going to bind to these reactive thiol groups of your lipoyl lysine residue. We're also now you may have noticed, we've taken this acetyl group and we've now converted it to a carboxylic acid. So we've oxidized this. If we oxidize a portion of the molecule, we have electrons that have to go somewhere. So you'll see with our sulfur groups, we've now added an electron here. So this is the reduced form of the lipoyl lysine residue. Now in enzyme 2, the CoA is going to come in. The CoA can pull that acetyl group off and form a high energy thioester bond here, releasing the now reduced lipoyl lysine residue. The reduced lipoyl lysine residue is now going to interact with the enzyme 3. FAD serves as a cofactor permanently bound. The FAD initially accepts the electrons from the reduced lipoyl lysine residue, forming FADH2 and regenerating the oxidized form of the lipoyl lysine residue. FADH2, now reduced, will donate the electrons to a free electron acceptor, NAD, producing NADH. This NADH can now travel to other reactions such as the electron transport chain or wherever is needed at the time. Now that we've produced our acetyl-CoA, we can move into the citric acid cycle. One thing that I want you to pay attention to as we go through the steps of the citric acid cycle is that we are never completely destroying a molecule. We're simply moving things through. I want you to think of the citric acid cycle kind of as a wheel that's always turning. How fast it's turning depends on all the other side reactions that are feeding into the citric acid cycle. So you can see here with acetyl-CoA, we have 2 carbons. We're going to combine with the 4-carbon oxaloacetate. So 2 plus 4 is 6. We get the 6-carbon citrate. We rearrange, forming isocitrate. Then spit out a carbon. We have the 5-carbon alpha-ketoglutarate. Spit out a carbon. We have the 4-carbon succinyl-CoA. Those 4 carbons then just need to have a few rearranging steps to succinate, then to fumarate, continuing to oxidize, fumarate to malate, malate back to oxaloacetate. So it's very key here that you see the purpose of the citric acid cycle is not to produce oxaloacetate. We started with oxaloacetate. The purpose of this cycle was to completely oxidize our carbon molecules, collecting the electrons in the form of NADH. A good way for you guys to study this is by first memorizing your 8 steps. And I tried to kind of summarize here so you could pay attention to what's really key in each step. All right, so let's go through each of our steps. Step number 1, we're going to form a carbon-carbon bond by combining acetyl-CoA with oxaloacetate. I've also boxed in here the 2 carbons that you're going to lose as carbon dioxide so that you can keep up with that as you go along. The major driving force that allows our citrate synthase to complete this reaction is the breaking of this high-energy thioester bond that was formed in the original Pyruvate Dehydrogenase complex. So you can see this is a highly energetically favorable reaction, and it's a condensation reaction. Step 2, our citrate is going to be simply rearranged a little in a dehydration reaction. Citrate will be converted into isocitrate. So you can see a water comes out, and then it shows you here boxed in orange. That's your molecules that form your water. And then another water comes in so that we can simply change the overall structure. The standard delta G for this reaction is pretty near 0. It allows us to decide which way the citric acid cycle needs to flow depending on which molecule you have more of at the time. So if the citric acid cycle needs to continue, the isocitrate will be pulled, allowing more citrate to be converted into isocitrate. In step 3, we get an oxidative decarboxylation. We lose our first molecule of CO2. Isocitrate is going to be converted into alpha-ketoglutarate. In step 4, we get our final oxidative decarboxylation step where we lose our last carbon dioxide molecule, you can see here, converting alpha-ketoglutarate into succinyl-CoA. With this step, we bring another CoA molecule in so that we are forming another high-energy thioester bond. Forming this high-energy bond is going to allow us to produce an energy equivalent in the subsequent reaction. This alpha-ketoglutarate dehydrogenase enzyme is actually a complex very similar to the pyruvate dehydrogenase complex. You can see here the steps are very similar. Main difference is going to be the size of your active site within your enzyme, where you're looking at a very small substrate with pyruvate versus a very large substrate with alpha-ketoglutarate. Continuing our steps, we'll now take the succinyl-CoA and convert succinyl-CoA into succinate by succinyl-CoA synthetase. This enzymatic reaction is very favorable because of the loss of the CoA molecule. This is the one step where we get substrate level phosphorylation in the citric acid cycle. A GDP molecule will come in and be directly phosphorylated, releasing GTP. GTP can then be converted directly into ATP by some processes that we'll see a little bit later as we go along. In our next step, we're going to oxidize the succinate into fumarate, turning an alkane into an alkene. This one step is going to be actually also part of the electron transport chain. So when we get to that section in this discussion, you need to make sure you recognize the enzyme Succinate Dehydrogenase. This particular enzyme uses FAD instead of NAD as the oxidized electron carrier to accept the electrons in the oxidation process. And that's going to come into play when we're looking at energy production later on down the line. From the fumarate, we will then use fumarase to produce malate. This is going to be a reaction that's a hydration reaction. We'll take our double bond, hydrate, so that we add an alcohol group on one side. Again, this is just a rearrangement step. Moving then, malate is finally converted back into oxaloacetate, so that we're taking the alcohol and regenerating the ketone group. This particular step uses malate dehydrogenase, and a final NAD is used to produce NADH. This is an unfavorable reaction. The only way this reaction continues is because the oxaloacetate is continually pulled by citrate synthase to combine with the acetyl-CoA to produce citrate, keeping this cycle going. Overall, our net result of the citric acid cycle, we're going to take that one acetyl-CoA molecule, release 2 molecules of carbon as byproduct, fully oxidized carbons. We produce 3 NADH, 1 FADH, and 1 GTP. So what we end up getting from this is, we'll have to do a little bit of math as we continue, but every time we produce an NADH molecule, every NADH is going to give us around 2.5 ATP. And that may sound a little crazy at this point. How can we make half of an ATP? But what we'll see is as we get to the electron transport chain, every time we have an electron moving through the electron transport chain, we are almost turning an enzyme that's allowing flow of protons that's producing ATPs. So what we're really doing with the NADH is getting about 2.5 turns out of that enzyme. So overall, we can go and do the math and say every time we have 3 NADHs, we get about 2.5 ATPs. Every time we have 1 FADH2, we get about 1 to 1.5 ATPs. Okay? So if we look at just the citric acid cycle, for every 3 NADHs, we'll get 7.5 ATPs. For every 1 FADH2, we'll get 1.5 ATP. So 7.5 plus 1.5 is 9, plus the 1 ATP from the GTP, we get 10 ATPs for every round of the citric acid cycle. Now if we go back to the original glucose, we actually got 2 acetyl-CoAs from the 1 glucose. So we would do the citric acid cycle twice for every glucose molecule. So for every glucose molecule, we get 20 molecules of ATP from the citric acid cycle. From these steps of glycolysis, we'll get 12 more. So overall, we get typically 32 molecules of ATP. So I really recommend that you sit down and see if you can do that math. So if we just think about it, here's the steps where we use or produce ATP or ATP equivalents. So we have 2 steps in glycolysis where we use ATP, but then we have a couple steps where we make ATP or NADH. Okay, so here is showing you your number of ATPs, and then your number of ATP and or NADH equivalents, and then here you get your number of ATPs ultimately formed so that we can do the math. One interesting thing about the citric acid cycle is that it serves as an amphibolic hub. So the citric acid cycle is going to complete the cycle for the purpose of producing reduced electron carriers, but also for the purpose of pulling things from the intermediates of the citric acid cycle to make other molecules. So the oxaloacetate can be pulled back out to produce molecules for gluconeogenesis. Oxaloacetate can be used to produce certain amino acids as well as nucleotides. Same for alpha-ketoglutarate. Citrate is used in the production of some of our fatty acids. So it's a highly convoluted process that we can look at. And that's really what makes metabolism, I guess, more difficult and challenging to learn, but it's also what makes it very interesting. Regulation of the citric acid cycle is pretty straightforward. We're going to see the key regulatory steps towards the beginning, because remember, these are the steps where we are doing the final oxidation steps of some of our carbons. So if we are going to stop, we want to stop before we completely oxidize those carbons in those highly exergonic steps. These steps over here towards the end of the citric acid cycle, if you remember, they were the steps that had very moderate delta G's. So those are not, I hate to say as important reactions, but those are not important as far as regulation occurs. The most heavily regulated step is going to be the conversion of pyruvate into acetyl-CoA, because there's no reverse there. So the pyruvate dehydrogenase will be inhibited by ATP and NADH and fatty acids. It's going to be activated by the opposite. The inhibition of pyruvate dehydrogenase by fatty acids is due to what we'll learn in our next chapter. When we break down fatty acids, we produce acetyl-CoA. The citrate synthase step is inhibited by NADH, succinyl-CoA, citrate, and ATP, the ultimate products, activated by a need for energy. Isocitrate dehydrogenase, inhibited by energy, activated by energy, or calcium signaling the need for energy. Alpha-ketoglutarate dehydrogenase, inhibited by NADH, one of the ultimate products of the citric acid cycle, but again activated if we have high levels of calcium, because that signals a need for energy.